Welcome to our first online session of Microbiology Lab. This is extremely weird for all of us. I've never had to teach online before, and none of you had to really learn online before, unless you've taken online classes. So, we're going to get through this the best that we can. I'm still figuring it out, you're still figuring out. I strongly encourage you to ask any questions if you might have them. Um, I'm here for you. I'm not doing anything at home other than sitting in quarantine and playing with my cat. So please send me emails. I am here for you to help in any way I can. So, this first little lecture that we're going to do that I'm posting is a continuation of Lab 15 that we did last time. If you remember correctly, we did Lab 15 and then I forgot to do the lecture for the continued part. Sorry, it was Lab 14, Exercise 14, and then Exercise 15 was a continuation. So today I'm going to go over the results for Lab 14, and then there's some information on Lab 15 for you with results. So let's get into it. So here is a table of the different organisms we did last lab before we were all dismissed from campus. Um, you'll see here on the left for Streptococcus pyogenes, it shows beta hemolysis, indicating that it is the complete destruction of red blood cells. Remember that, it's very important that you remember that beta is the complete destruction of those red blood cells on that blood agar. It's sensitive to bacitracin or the A disc, and it is resistant to optokin or the P disc. So keep in mind while we're going through these results, excuse me, keep in mind what sensitive and resistant mean. What does that look like on a plate? You all have done enough antibiotic discs on different plates for different tests. I have I have a lot of trust in you that you know what resistant and sensitive look like by now. Okay, so now Streptococcus agalactiae, that also exhibits beta hemolysis. It was resistant to bacitracin or the A disc, and it was also resistant to optokin or the P disc. Next was Streptococcus pneumoniae. It exhibited alpha hemolysis. Remember, alpha hemolysis was the incomplete destruction of those red blood cells on the surface of that the blood agar plate. It was resistant to bacitracin, but it was sensitive to optokin or the P disc. Next, we had Streptococcus mitis. It also exhibited alpha hemolysis. It was resistant to both the A disc and the P disc, being bacitracin and optokin. The last one we had was Enterococcus facialis. It exhibited gamma hemolysis, which recall back to the last lab we had. Gamma hemolysis means that it does not break down those red blood cells, right? It does not break down red blood cells. There is no hemolysis taking place. So it was also resistant to both bacitracin and optokin. And this next slide here is just a reminder to what the different results look like when I say alpha, beta, and gamma. Remember that alpha is the incomplete destruction of red blood cells. You have that kind of greenish color. That's the clearing that it exhibits on the blood agar. The beta hemolysis is the complete destruction of the red blood cells on the agar, and you can see by the picture here that the zone of clearing is a lot more pronounced, and the red has just gone away. Sorry, I don't know if you could hear that. I just got an email. So if you probably didn't hear it, but never mind. Anyway, and then the last one we have is gamma hemolysis, which, as we know, does no it has no hemolytic qualities. Therefore, it stays on the surface of the blood agar, and it does not break anything down. Okay. So this next bit of information is the stuff that I forgot to mention in the last lab. Oopsie daisy, that's my bad. I'm very sorry for that. But it's all good. Uh, you guys will have it now. So this was the stuff I forgot last lab. Um, you did both of the tests for this lab. We just weren't able to look at the results, and I wasn't able to tell you the importance of these two tests. So I'm going to do that now. So the two tests that you guys did were the bile escalin and the sodium hippurate tests. And why we do these tests is because this group of streptococci called group D, they can be distinguished amongst each other because of their hemolytic qualities, but they can also be distinguished by their ability to hydrolyze escalin into escaletin. So it's a enzyme kind of thing happening here. It's something's going from escalin, breaking it down to escaletin. Escaletin reacts with ferric ions or iron ions to produce a black product. This test is useful for distinguishing the viridins group of alpha hemolytic streptococci and Enterococcus facialis. So this test is very useful in distinguishing between the two groups that we're looking at right here, alpha hemolytic streptococci and Enterococcus facialis. So this next slide is just showing you how this kind of works. So we have escalin, and it gets hydrolyzed into escaletin. And because we add these ferric iron ions, or the ferric iron ions are present, it creates a black product in the test tube. And this black product is how we know that the organism present is positive or negative for the bile escalin test. If it turns black, a blackening of the auger slant, if that occurs, that means it is positive. So now let's talk about sodium hippurate. So the sodium hippurate test helps us to differentiate Streptococcus agalactiae, or group B Streptococcus, from other beta hemolytic streptococci. And what this test tells us is if something has the ability to hydrolyze sodium hippurate to sodium benzoate and glycine. So it tells us if something has the ability to hydrolyze sodium hippurate to sodium benzoate and glycine. And the way that we tell if this is occurring is by using the CAMP, or the CAMP test. And a little bit more about the CAMP test. It was named after these group of scientists, Christy Atkins and Munch Peterson, and it differentiates group B streptococci from other beta hemolytics. It's done on a blood agar plate, just like our hemolysis. That's what it is. Just like those, they're done on a blood agar plate. And what you do is you streak Staphylococcus aureus. You streak it perpendicular to a streak of beta hemolytic streptococci on the plate. So you have a perpendicular streak of Staphylococcus aureus, and then you streak a beta hemolytic streptococci onto the plate. And I'll show you a picture of this. I have a pretty good picture in the following slides that'll explain this a little bit more. But if the CAMP test is positive for group B streptococci, like for example Streptococcus agalactiae, we will see a large area of red blood cell lysis forming at the juncture of the bacteria. So where the two bacteria are meeting, where Streptococcus agalactiae is meeting with Staph aureus, we're going to see an area of clearing. And that'll be evident in the picture I'm about to show you. and it looks like an arrowhead. Red blood cell lysis occurs as a result of the beta hemolysis enhancement due to the beta lysine that is given from the Staphylococcus aureus and CAMP factor. And CAMP factor is just a peptide that is only formed by group B streptococci and listeria species of bacteria. So this example of streptococci, the Streptococcus agalactiae, has the CAMP factor. So it's a peptide only formed by the group B streptococci. So that means that we can differentiate our group B Streptococcus agalactiae from another beta hemolytic organism. Okay? And so this next slide is just a picture showing the results of a CAMP test. You'll see that that the big streak down the middle, that's Staphylococcus aureus, okay? So that is Staphylococcus aureus, and then on either side that are labeled A and B, we have our different species of Streptococcus. So A is our Streptococcus agalactiae, and B is a negative Streptococcus pyogenes. Okay? And what it's showing here, you can see where A meets that long streak of Staphylococcus aureus. It has that arrowhead kind of formation right there, that arrowhead of clearing. That is because of the cyclic AM, or sorry, the not cyclic AMP. It's because of the CAMP factor that Streptococcus agalactiae offers, and that's also because of the beta lysine offered from Staphylococcus aureus. So that's why A looks like that. That's why that arrowhead forms right there, and that's also why B is negative. Staphylococcus aureus is still offering this beta lysine, just like it is to Streptococcus agalactiae. However, B is not an organism that exhibits this CAMP factor. Okay, so that's why it's negative, and that's why A is positive. So this next slide is just the results that you would have seen from your bile escalin and sodium hippurate tests. Obviously, we can't get into the lab and look at the slides ourselves, so I've attached some pictures that will be helpful, and we'll talk about the results. So for Streptococcus pyogenes, for bile escalin Streptococcus pyogenes, it was negative, and for sodium hippurate, it was also negative. For Streptococcus agalactiae, for bile escalin, it was a negative result. For sodium hippurate, it was a positive result. For Streptococcus pneumoniae, it was negative for both. For Streptococcus mitis, it was negative for both. And for Enterococcus facialis, it was positive for bile escalin, and it was positive for sodium hippurate. And what positive and negative means, you guys can look on page 64 of your lab notebook. It'll help you a little bit. A positive bile escalin test is a blackening of the agar slant, and that's after incubation. So a positive will be black, a negative will not. It'll look like nothing changed after incubation on the slant. For the sodium hippurate, if it is positive, after incubation, we would have added 3 drops of this reagent called ninhydrin. We would have added it. We would have waited for a good 5 minutes or so, and then we would have been able to read and record our results. And a sodium hippurate positive is a purple color will form after we add this ninhydrin reagent. So if a purple color forms, it's positive. If no color forms, it is negative. So this next slide is just showing you the difference between a bile escalin negative and a bile escalin positive test. The bile escalin tube on the left there, you can see no color change has occurred after incubation, so we would call that a negative test. However, the bile escalin on the right is blackened after incubation, and that is indicative of a positive test result, right? Because we mentioned how that When we have escalin being hydrolyzed to escaletin and these ferric iron ions come into play, we have a black product formed. And that's what that is indicating. The black product formed, meaning escalin was hydrolyzed to escaletin, ferric ions played a role in changing the color. And this last slide here is just showing you the different results for sodium hippurate positive and negative test after adding our ninhydrin reagent. And what this shows is on the left we have the color change to purple, indicating that a positive test has occurred, and on the right we have the negative test. That's all I have for this one. The next video will be exercise 16, differential media for enterics. And then the one after that will be exercise 17, multiple test media, okay? So that's all I have for this one. Please feel free to ask questions. I'm here for you guys. Here to help in any way I can. Reach out to me, send me an email. Even if you have questions about anything. If you have questions about coronavirus, I'll do my best to help you out. I'm no expert, but I know a little bit about it. So yeah, stay safe out there, and I will continue to talk to you guys and hear from you. Alrighty, bye-bye.